Okay. Yeah, absolutely, Daniel. We can definitely do that. So we'll review the bonds. And if as we're going through today, if you think of any specific questions relating to bonds, we can absolutely talk about those. Or if you just want me to review bond structure, the kinds of bonds, we can definitely do that as well. All right, so pull those things over there. Cool. So chapter 3, this is like kind of our last little homage to our high school curriculum. And we're going to talk about the cell, right? So we got to understand the cell, because if we're thinking back to kind of our hierarchy of matter, we're moving up the chain here. So we're starting to think about, okay, we've already got our foundational knowledge for the atomic and molecular basis of things. We've got a pretty solid foundation of chemically what could be going on in the body, but what kind of an environment is this sort of stuff happening in? Well, the smallest environment that these things can happen in is either within the cell, or sometimes it happens outside the cell. A lot of the time, all of the chemical reactions that we're talking about are happening either inside the cell or immediately around the cell. And we'll talk about why the structure of the cell itself is really conducive to things like that. So our goal today is to kind of, strike out on this kind of flyover journey of what the cell looks like in general. And the funny thing is the image of the cell that you'll see is hardly ever what an actual cell will look like underneath the microscope in an anatomy class that you'll see. There are some cells that'll look like it, but for the most part, a lot of our cells are going to look like the configuration of epithelia that we talked about in lab last week. So that's kind of the funny thing about like the classic cartoon image of what an animal cell looks like. But we're going to talk about what a generic animal cell looks like. We're going to talk about some of the components that these cells have, so some of their organelles or the organs of the cell. Talk a little bit about what those things do, and then if not today, then probably on Thursday or our next lecture, we'll start to dive into some more of the kind of broader mechanisms that cells are responsible for. So things like DNA replication, cell division, going from an image or going from a sequence of information on the DNA code to a fully functional protein, or the process of transcription and translation. So that's what we'll end up getting to at the end of our chapter 3 material. Okay. So funnily enough, in your book, this image is dealing with a child who, or showcasing a child rather, who has a condition known as diabetes insipidus. So, okay, for sure. Thanks, Sean. I understand, hey, work is a thing, so no worries, dude. So with this figure, if you look at the description for this in chapter 3, if you have the book next to you, it's talking about a child that has diabetes insipidus. And for those of you who aren't familiar with it, or you don't want to bother with a Google search, diabetes insipidus is a condition where your body is unable to regulate fluid levels. And so what ends up happening is some of the side effects are, you can have just overwhelming sensations of thirst, and your body will produce really large volumes of dilute urine. And so that's part of the problem is that wreaks havoc on the kidneys, right? And in essence, some of the pathology of diabetes insipidus is related to improper kidney functions, but it can also be neurologically related. It can also occur during pregnancy. There's a whole bunch of reasons why diabetes insipidus can happen, but this image talking about fluid regulation and homeostasis, I feel would be better suited for something like chapter 2, but here it is in chapter 3. But the way that we can relate this to some of our chapter 3 material is kind of at the end of this chapter, we're going to really understand how cells on their own are important and why they can be put together to form complex different layers of tissues and then ultimately organs. So that's kind of our ultimate goal here, is to understand why something like the kidneys could be functioning improperly on something like a cellular level. Right? Okay, so here's our hilarious cartoon image and cartoon representation of what a cell looks like. So when you guys look at this, I don't want you to think about necessarily that this is exactly what an animal cell looks like, right? Because you already know that that's not necessarily the case. You already know this from, from lab last week, that really what this image is telling us is it's an idealistic representation of all of the stuff that a cell can have. And for the most part, all of our cells in our body have most of the internal components here. The external components that we're seeing out here are where a lot of the variation occurs, and also some of the structural components. So like this is kind of a large globular cell, and some of our cells can look like that, but we also know that cells can take on different forms. We know that they can exist in these cubes. We know that they can exist in columns. We know that they can exist in layers, right? So when you look at something like this, don't necessarily think of it as this is what a cell looks like, right? This is just a idealistic representation of everything that a cell can have. So let's kind of go through some of these things. One thing that we're going to talk about in detail is the cell membrane. This barrier that encloses the cell is very important because it controls what goes in and out of the cell, for one thing, but it's also going to control most of the physiological mechanisms in our body because it's really good at creating these artificial concentration gradients. And so if you guys remember, we talked, I might have talked about this at the beginning of chapter 1, but if you understand what a concentration gradient is, okay, if you understand what this is, which, and that's, we'll try and get to this today. Those two words can explain literally every single physiological mechanism in the body. Here, let me scoot this forward. I realize it's like a little bit far back. That might be a little bit better. There we go. So concentration gradients in a definition form would just be where you have a difference in concentrations, right? And usually the best way to think about this is like, if you have a high concentration of something on one side of the room, and if you have a low concentration of something on another side of the room, eventually those two particles will equal out in distribution due to other movement mechanisms that we'll talk about as well. Another good way to think about this too is like, so for example, if we were all to be in a lecture hall, if we were to be in Canyon Hall 118 right now, which is where this class should have been, and those of you who have been on craft and hopefully you guys know where that room is. It's a pretty good sized lecture hall. It's one of the bigger ones that I've taught at craft in, but it's nice because there's a lot of space for everybody to sit in, right? So it can fit a lot of people. But the cool thing about our classes in general, we don't typically fill up that lecture hall, right? So what do you do if there's not enough people to take up every single seat? Do all of you sit on one side of the room right next to each other, right in everybody's space? No, of course you don't. We all leave. It's an unspoken rule. You leave that one chair in between each person so everybody's got their bubble and everybody's happy. Nobody's sitting right next to you and nobody's right in your space, right? Like that's what we do. So we already have this natural sense of we don't want to be all packaged into one area. We want to be spread out. We want to be nicely spread out. So molecules behave much the same way, but if they get spread out too quickly or too evenly, then we can't use those molecules to do things like cellular work. So what our cell is really good at doing with something like the cell membrane is the cell membrane provides a nice barrier to where we have other components that can force molecules to one side to where they don't want to be. And then the cell membrane kind of acts like this door guard that says when those molecules can and can't leave. And that's nice because we can control the movement of that and we can use that to do things like cellular work. So in essence, those are some examples of what concentration gradients are. And if that doesn't make any sense right now, that's okay. We'll circle back around to that. But I just wanted to preview that because a lot of the other mechanisms that we're going to talk about in this chapter and virtually everything else in this class can be boiled down to an understanding of concentration gradients. Okay, so the cell membrane is nice and important, or is nice because it's important for helping control these concentration gradients. These little sticky outy bits, these cilia and microvilli, there's a couple instances of where we've seen these. So remember, cilia are little hair-like extensions that don't contain any components of the inside of the cell membrane. So all the fluid that's inside of the cell in here, all this yellow stuff that you're seeing, is a fluid called the cytoplasm. Sometimes it's called the cytosol, depending on kind of, if you're talking to a cell biologist or an anatomist or whoever, but it's all fluid that makes up the inside of the cell. So importantly, the cilia doesn't contain any of that. It's literally just like a little hair-like extension. It's closed off, it's its own thing. But the microvilli, on the other hand, are important because they do contain small portions of the cytoplasm. And depending on the situation, they can actually contain cellular components like proteins and things that are responsible for transport in and out of the cell. And that reason is to enhance something that we call the surface area to volume ratio. So that's one of our other important concepts to know. So surface area divided by volume. So that fraction is what we call the surface area to volume ratio. And that ratio is important because it determines things like cell size. So it's what dictates the fact that an elephant, a mature African bull elephant, has the exact same size cells that a field mouse does. It just has more of them. So this is why things like really large animals on this planet can only have a certain size cell. If they get too big or too small, then they can't perform the same metabolic activities that they would normally, right? So cell size, so how much surface area it has relative to how much volume or 3D space that it takes up is important. And the way that we can kind of cheat some of the physical restrictions that cell size has on our body is we can create these little folds in the cell membrane here. And so these little villi-like extensions are nice additions to increase the amount of surface area that we have relative to the amount of volume. Another way to think about that would be like if you take a big rock and you smash it up into little pieces, that rock, so that big rock and then all the little pieces that you made from it still has the same volume, but the amount of surface area that you've introduced is much, much greater. So let's say that rock has 6 sides and you split it in half, okay? Then you've now made 2 cubes that each have 6 sides, but they still have the same volume, right? So now instead of having the same amount of surface area, or so like 6 times 6, or no, I'm sorry. So if we do like a quick math calculation, so if we've got our cube, this is a really bad cube, I know. Okay, but let's say that each one of these sides has a length of 1. Don't worry about units, that's arbitrary. So the way we calculate area is we do length times height, right? So 1 times 1 is 1. But if we're talking about something like surface area, so surface area is the number of sides times their area. So times the individual side area. We have 6 sides, but each one of those sides has an area of 1. So 6 times 1 equals 6. So the total surface area of this cube would be 6, if that makes sense, okay? So then when we talk about surface area to volume, the way that we calculate volume is we just add the depth dimension. So again, that's also going to be 1. So the volume of this cube is length times width times height, 1 times 1 times 1. So the volume is going to be equal to 1. Okay, so volume is equal to 1. So then if we put those values in up here with our surface area divided by volume, so 6 over 1, you get a value of 6, okay? So having as high a number as possible is really important for kind of cheating the limits of our universe, right? So to put that in perspective, let's say that we change the side length of each of these sides instead of 1 to 5. So if we change that to 5, okay, so 5 times 5 is 25 times 6, okay, is, I don't know, let's do quick math, calculator, here we go, 25 times 6, so 150, okay, so for total surface area. So 150, so 25 times 6, or no, 6 times 25, the way that I have this written. So 150. But then if we calculate the volume of this cube, so 5 times 5 times 5, that's 125. So volume now okay, so volume now is 125. So 150 divided by 25 is a much smaller number than this one. So believe it or not, the cube that only has side length of 1 versus the side length of 5 has a much larger surface area to volume ratio. The larger the number, the better it is for living systems. And we'll come to understand that a little bit more later. So here, actually, let's, so then we'll complete our math here. So surface area to volume. So 150 divided by 125 is equal to what? So 1.2. Okay. So the rationale, the understanding behind that will come with the rest of this chapter. But thinking about things in a simplified mathematical form like this is helpful for us, because then we can understand a little bit more why our cells are the same, or why our cells are only a certain size, why our cells are pretty much the same size as they were millions of years ago, right? Because we reach the upper limits of how big they can be to sustain the kind of metabolic activity that they have. And we'll find out that that's the limiting factor, is whatever the metabolism of the cell is, if it's got a higher metabolism, it can only get to be a certain size. If you've got a really slow metabolism, then you can probably be a lot bigger. But we as endotherms, as mammals, a lot of our energy that we consume and that we don't generate, but that we transform from our diet, is utilized in maintaining things like temperature. And so that's limited by cell size. Okay, so I kind of got off on a little bit of a tangent there, but I promise you these are not going to be things that, oh, you won't be tested on that, right? These will show up in one form or another. So understanding concentration gradients and surface area to volume are important, right? So if you don't understand it now, that's okay. We'll get into that more in a little bit. Some basic functions of the cell, like I said, metabolism. So remember, the cell is the most basic unit of life, and each of these things encompass the basic necessities of qualifying something as a living thing. If you are a cell, then you have a metabolism, whether it's a fast or slow metabolism, or a relatively large or relatively small metabolism, you have a metabolism. So that means you are taking in nutrients, and that means you are expelling waste in some form or another. Cells reproduce as well. So this cell can split itself in half and make a clone of itself, or depending on where we find it, they can be found in sex organs, like, or actually not the sex organs, but what's the term? I'll use sex organ, but it's not the technically anatomically correct term. But organs like the testicles and the ovaries, which produce sex cells or gametes. Okay, so the genitals, that's what I was looking for. So those organs produce cells that only have half of the amount of DNA that something like this would, right? With the ultimate goal of fusing with another cell in the form of an egg or a sperm, depending on the perspective, to create a new life form through, instead of asexual reproduction or clonal reproduction, you do that through something like sexual reproduction. All cells can communicate in one form or another. So what we'll end up talking about, and why things like surface area to volume ratio and concentration gradients are important, is these are some of the ways that cells communicate. Cells don't have verbal communication. They don't have eyes, they don't have ears, they can't smell anything, they can't speak, they don't do any of those things, right? So how do they communicate? Well, they communicate based on molecules and how much of a particular molecule that they have. So if a cell is producing a ton of a particular molecule, that molecule will probably be expelled out of the cell through, something like a phagocytic vesicle over here. And then those molecules will go on to send a signal to some other cell that's usually right next to it, right? Or if there's channels that are linking one cell to another, those molecules can move from one cell to the next. Well, how the heck do those molecules get there? Through concentration gradients. Just like humans sitting right next to each other in a lecture hall, molecules do not physically detest it. They hate being crammed in the same space as something else that is similar to it. So what do? Well, they bounce off of each other and create a lot of kinetic energy due to high concentration. And then due to those random interactions, they will naturally move from high to low concentration. That's our definition of our concentration gradient. That's how we control the movement of chemicals from one area to the next, right? That's one of the main ways that cells are able to create communication channels between themselves, and then subsequently, depending on the impact of whatever the chemical is, can have huge impacts on other organs and other organ systems. So for example, if we kind of refer back to diabetes insipidus, one of the reasons that can occur is through a neurological disorder that affects the pituitary glands. You can have a tumor, you can have a developmental issue, whatever, but you hinder the production of a hormone called ADH, or antidiuretic hormone. It's also called vasopressin. And I think we talked about it before in one of our other lectures too. But that chemical, that hormone, which is produced in the brain, affects the functionality of the kidneys, which are a completely different part of the body. But they're all produced by cells that are in the brain that affect cells that are in the kidney elsewhere in the body, right? So depending on what the molecules are, they can have what's the phrase? They can have widespread effects on the body, or they can have small localized effects too. It just kind of depends. And that's kind of the cool, interesting thing about cells and the molecules that they produce, is just depending on what they are, they can have, small or large effects. So kind of that goes into the last point here with synthesizing molecules. Most cells are not just going to sit there and do nothing. They're not just going to sit there and metabolize everything and then produce waste. A lot of them are going to be contributing to larger tissue and organ functions by producing molecules. And a lot of those molecules get produced in the form of proteins. So all of that is going to happen right around here with the nucleus, and then this large kind of crazy cavernous structure here called the endoplasmic reticulum, right? So cells are really cool and they're really interesting, and they do a lot of really neat things. These four basic components here are both the primary functions that you need to classify something as biologically alive, and they also happen to be the basic functions of cells and what cells do. Okay. All right. So here we've got a nice little table that breaks that down for us too. So what are these organelles and what do they do? So the nucleus, kind of back here, So the nucleus, this big old purple sphere looking thing, right? It depends on the cell. Most of the time it's in the center, but depending on what kind of cell we're talking about, it can get pushed out to the side. But it's usually near the center of the cell. Contains most, not all, but most of the genetic material in our cells in the form of DNA, or deoxyribonucleic acids, and nucleoli. So these are the sites of RNA synthesis and ribosomal subunit assembly. So if you kind of remember back to chapter 2, at the end when we were talking about our macromolecules, we talked a little bit about nucleic acids and their role as a macromolecule. Nucleic acids are the code that makes us look the way that we do and act the way that we do and behave the way that we do. So all of that information being wrapped up in DNA has to be expressed somehow. And that's the purpose of RNA. So RNA and ribosomes take that information from the DNA and actually use it. They convert that information into usable things, right? The rest of the cell doesn't give a crap about what the DNA is saying because it can't understand it. DNA is this like wise old sage that speaks some crazy language that nobody understands, but we know that he's really smart. So we have people that can interpret that, the ribosomes and the RNA, they interpret that information and make a functional, useful thing out of it. That the rest of the cells like, oh, cool, like we can actually use this thing. We don't know what the heck that crazy dude's talking about, but you guys got this from what he was saying? That's sick. Let's actually use that to make the cell function and work the way that it should. So that's the purpose of those things. Ribosomes, okay? So ribosomes are these little kind of floaty guys. Sometimes you have them floating around in the cytoplasm, like these little black dots here. But sometimes they're embedded within the endoplasmic reticulum right here. So you can see all those kind of little dots right there. The whole purpose of a ribosome is to make protein. So ribosomes themselves are not considered proteins, but they are a blend of RNA and amino acids, but they're not considered functional proteins, but they're a blend of those two things. So RNAs are these cool little kind of Pac-Man looking molecules where they essentially chomp down onto a little strip of RNA, which you can think of as like, it's like a transcript from the people who were listening to what this crazy old sage guy was saying. They're like, all right, here's what he's saying. Let's send this out to the factory and let's have them make this thing. And the ribosomes are like the little factory workers. They're like, all right, cool. We're going to read this blueprint and then we're going to make a product out of it. So that's what they're doing. The rough endoplasmic reticulum is all of this stuff that is stuck right up against the nucleus here, right? So the entire endoplasmic reticulum is all of this. So this entire thing right here, all in this kind of pinkish, bluish color. The blue is the inside and the pink is the outside, just to kind of show that there's a difference between the two areas. So all of that that's wrapped around the nucleus right here is the endoplasmic reticulum. But there's rough portions of it that have the ribosomes stuck on it. And then there's smooth parts of it that don't have the ribosomes stuck onto it. So now that we know that ribosomes are used for making proteins, the rough endoplasmic reticulum, or the rough ER, probably has a lot to do with protein synthesis. Okay, has many ribosomes attached, site of protein synthesis. Cool, right? The smooth portion doesn't just sit there and do nothing because it doesn't have any ribosomes. The smooth portion is going to produce lipids. So now if you recall from last week when we talked about lipids or fats, remember those things are built from hydrocarbons. So they are made out of fatty acid tails, okay? Let me close the door really quick. All right, so those lipids are important because they are going to help with maintaining the health of the cell membrane, right? The cell membrane is quite important. So if that's not clear now, you know, it should be getting to that point. The cell membrane and the health of the cell membrane is crucial for the cell being able to perform all of the things that it does, right? The smooth endoplasmic reticulum helps with maintaining the health of the cell membrane. And it also helps when we decide to, you know, have that one extra beer or that one extra glass of wine or that extra shot, right? When you introduce all these toxins into your body, one of the ways that our bodies can process those things is within the liver primarily, which has cells that have these portions of smooth endoplasmic reticulum that can help process and detoxify some things that we introduce into our body. It's not just alcohol, it's other toxins as well, but the smooth endoplasmic reticulum helps with that. The Golgi body. Okay, so the Golgi body is this green guy that's sitting out right here that has all these little bubbles kind of floating around it. The Golgi body is kind of cool because it's like a USPS service that doesn't have any shipping delays, right? I don't know if any of you guys have experienced that lately, but my wife's birthday was yesterday, and so I had ordered, you know, all this stuff like a week and a half ago, And I'm glad I did, because there was like a massive shipping delay, and it's going on right now with the postal service and everything. But the Golgi apparatus, its whole purpose is to fold and package and ship out the proteins that the ER makes, but it does so in a way that it can modify them if it needs to. Sometimes the proteins that get produced by the rough ER are not like fully finished products, or maybe they were finished initially, but then we get a little message saying, all right, crud, we forgot to add this thing on, okay? The Golgi body takes all those and says, all right, cool, I got this. I'm gonna send this protein to this part of the cell membrane, and it's gonna go on this side. I'm gonna send this protein over here. It may get stuck to the mitochondria, or it might go back into the nucleus. I'm gonna send this protein over here that's gonna be expelled out of the cell, but before it goes, I'm gonna modify it with a little bit of sugar so that once it gets out there, that protein is able to interact with other cells and perform other functions. The Golgi apparatus modifies and packages proteins to be able to be shipped out to different parts of the cell or be expelled altogether. The other cool thing about the Golgi body is that it can produce these things called lysosomes, which are these bubbles, or what we call vesicles. So that's the next component right here, secretory vesicles. But a vesicle, is, think of it as like a little bubble of lipids that will protect a compound that's inside of them from the polar environment of the cytoplasm. Okay, so the insides of cells are water-based, so they're polar. Some molecules don't like to be in those environments, and some do. So the Golgi just packages everything so that it protects those things from getting destroyed in that polar environment. And the one way that it ships those things out is by producing these little fatty bubbles that we call vesicles. So secretory vesicles can contain materials that are produced in the cell, typically formed by the Golgi apparatus, and they get expelled or secreted out of the cell through a process that we call exo, meaning out of, cytosis. So getting rid of something from the cell, cytosis, cellular process, right? Lysosomes. So lysosomes are vesicles that just contain specialized proteins for breaking stuff down. Sometimes, if you're a plant cell, sometimes your chloroplast or your mitochondria or, something else in the cell is not functioning and needs to be broken down and recycled. Lysosomes can help do that. Cells got to eat. Cells need to be able to take in nutrients and process, food to give themselves energy to satisfy their metabolic requirements. Lysosomes are utilized for that. So lysosomes are kind of like just the generic processing feature of the cell. Peroxisomes. Peroxisomes are pretty cool because they're kind of like the hazmat waste crew. Peroxisomes can break down things like fats, amino acids, and more importantly, things like oxygen-based free radicals, or what we call reactive oxygen species. I'm going to put that in the chat up here. So reactive oxygen species. So they're also sometimes referred to as ROSs. So ROSs are interesting because they've got a lot of links to things like cellular health. So if you have an ability to be able to process a lot of reactive oxygen species, we're seeing that there seems to be a positive correlation with things like lifespan. So as you get older and as as we all go through life, our bodies are exposed to reactive oxygen species on a regular basis. And one of the reasons that they're an important focus on cellular health is because they can tear apart cell structures. Reactive oxygen species can actually destroy things like cell membranes, and that's not good for cell health. So if you possess the ability to break down a lot of these things that sometimes are just byproducts of the metabolism that we have, if you can break those things down effectively without it damaging the cell, then the cell can live longer and everybody's happy, right? So what we're seeing is that as we get older and as we shift, or as our metabolisms kind of shift and change with age, the ability to break down and process ROSs seems to be correlated to longer lifespan. A good example of this too is something like exercise behavior is people with a lot of slow twitch muscle, or muscles like you can think of people who train for endurance races like marathons, or just endurance running in general. people will typically have a greater percentage of muscle fibers that can break down reactive oxygen species that occur during exercise, like lactic acid buildup in muscles. So the ability to process those things more effectively is positively correlated with lower muscle fatigue, or I should say it's inversely correlated with muscle fatigue. The better ability to break that stuff down, the more energetic your muscles feel, right? So peroxisomes are pretty cool. Mitochondria, it's the powerhouse of our cell, right? That's our our new meme now, right? Mitochondria, singular mitochondrion, which is what we have here, is the site of aerobic respiration and the major source of ATP production within every single one of our cells. Remember, ATP is important because it's the basic energy molecule. That is the money that controls the economy of our cellular environment. So the more ATP you can produce, more cash you got in your pockets, right? So more ATP is good. Microtubules and centrioles, okay? So these are kind of built out of the same stuff. They're built out of like little small protein fiber components. The microtubules their main purpose is to kind of support the inside of the cell. They kind of serve as like an internal skeleton of the cell. So when it says supports the cytoplasm, it's kind of what we're referring to. It can also assist in parts of cell division. So when the cell is going to divide itself, the microtubules can kind of help the cell proceed with that reaction. But then if we take things like sperm cells that have the long tails that help propel them through different kinds of fluid, that the inside of that tail is composed of these long protein structures called microtubules that move back and forth just like a fish fin. And kind of at the base of the head of the sperm, there's a section there that has a ton of mitochondria that burn through ATP very, very quickly to power that movement. Okay. And then cilia, right? So microtubules, centrioles, and cilia. Actually, no, I've skipped centrioles. Centrioles are specific components or specific collections of microtubules that are built into much larger components that are utilized to pull the chromosomes apart during the cell division process. So remember, when cells divide, they kind of just split everything in half. One of the ways that we can make sure that we split our DNA in half is parts of the DNA get attached to these centrioles here, and that helps move them to the other sides of the cell. So cilia, on the cell surface with many on each cell, move substances over the surface of them, right? So we talked about cilia in tracheal tissue, so the lining of our windpipe. These cilia are quite long, and their whole purpose is so that we can make sure that we can expel or cough out any sort of foreign projectiles or bacteria that might get into our throat. Flagella, you typically only find these on sperm cells. To my knowledge, I don't think that there's a cell other than sperm that have flagella, in humans rather, that occurs in us naturally and isn't some sort of bacteria or something. But yeah, the flagella on sperm cell surface with one per cell helps move it through fluid. Microvilli, just like I mentioned earlier, extensions of the cell surface with many on each cell, increases surface area of certain cells. So these we'll find a lot within the intestines. So once we get to the gastrointestinal system, we'll talk a little bit more about that, but those are very important there. Okay, cool, so we're making good time here. Okay, the cell membrane. So this picture tells us a lot of information, all right? All of these little blue beads right here. So if you guys think way back to last week when we were talking about some of the structures of our fatty acids and of our lipids, we can make fats that can exist in both polar and non-polar environments. So everything that's out here on either side of the cell, so out here and then inside the cell down here where it says cytoskeleton, both of those environments are water-based. So, and water is polar. So lipids, which are mostly fats made out of hydrocarbons, So a bunch of carbons all linked together with hydrogens stuck to them. Okay. So this is the major portion of our fat. These things that are made out of hydrocarbons hate being in polar environments. They do not like it. Okay. The reason being is because this molecular structure here is not conducive to existing in an environment where there's a bunch of charged particles roaming around. Remember, water, so we'll draw the molecular form of water. So water looks like this in its molecular form. And the reason that those bonds aren't straight out is because we've got these little groups of electrons over here that are forcing those bonds down at this angle. And remember, the reason that water has its polar nature, first of all, is because of this bond. So remember, this bond that's binding oxygen and hydrogen here and here, this is a covalent bond, but more importantly, it's a polar covalent bond. Okay. So because of that, this area of our water molecule has that net negative charge. Siri's going to try and calculate polarity here. Oxygen has that net negative charge, and then right around the hydrogen atoms, these guys are going to have that net positive charge. Because those electrons are more closely associated with oxygen here. So by our understanding of molecular anatomy, and then by definition of a polar covalent bond, we create a polar molecule. And if you have an environment that's completely composed of polar molecules, then you create a polar environment, right? And hydrocarbons that are not polar, so all of the electrons here are even and they're happy and they're happy with just being like this. They cannot exist in a polar environment. They will be physically repelled by that. So then how the heck do we have a structure of a molecule that's got all these little fatty acid tails here and then all these little blue beads on top? Well, what our cells have geniusly evolved towards is having a structure of a molecule that has both a polar and non-polar part to it. So if we have something called a phospholipid, which is what each of these things are, we have our big blue bead. Let's see if I've got blue here. So we've got our big blue bead, and then we've got our little fatty acid tails. Okay. So this blue part up here, its major functional groups that make it unique are phosphates, which is where it gets the name phospho, and nitrogen, both of which are polar. These are polar elements. But then we've got our hydrocarbon chains down here that are nonpolar. Okay. So this is nice because we've created a molecule that has duality associated with it. We can now put this thing in both a polar and a nonpolar environment. So this phosphate nitrogenous head up here will orient itself towards polar environments, and the fatty acid tails will orient themselves away from it. So the cell membrane has two different environments associated with it. Outside, externally, and then internally on the cellular side is polar. So externally. And then actually, here, because I'm going to use the word internally again, so we'll say cell side. But then if we look at this figure, we see that we have two layers of these phospholipids, all right? So then if we've got another phospholipid down here, okay, we've created this interesting space in between these two molecules. I think you guys can see that. Okay, cool. So you've created this space in between these. So this internal space is now a non-polar environment because it's only made up of fatty acids. That's all that makes it up. So the cell membrane itself has a sense of duality because of its anatomy. So this is a key example in this class of form-function relationship. But we haven't talked about the function part yet. But the form, this molecule is structured a specific way. Chances are it's going to have a specific function. So let's talk about some of those specific functions. Because the beauty of this bi-layer, where we have two separate layers of these phospholipids right here. So the cell membrane, by definition, is something that we call a bi-layer. So if you're taking notes, that would be something to write down. Where we can create these dual environments, where we can create an environment that we've got a polar set of molecules that can exist and be completely happy on either side, so inside the cell and outside the cell, but then inside the membrane, inside the anatomy of our membrane, that is a separate environment that is a non-polar environment. So already, what might be kind of piquing your curiosity here is we can see these giant globular, like cool colored things sticking into the cell membrane here. Those are proteins. Each one of these big old globular things is a sequence of amino acids that is all folded up in a very specific fashion. And some of them are shoved all the way through the membrane and connect the polar external environment to the polar internal environment. Some of them provide a receptor-like molecule, kind of like an antenna, that can respond and receive other molecules that it may come in contact with. Some of those proteins exist only on one side. Some of them exist only on an internal surface. Some of them exist only on an external surface. These proteins can be embedded within various parts of the cell membrane. So now what you should be kind of thinking about is like, okay, well, how the heck is that possible? How can you have a molecule that can exist in both a polar environment outside the cell? Because remember, all of this white space out here, this is all water, and all of this stuff inside the cell is water-based as well, both polar environments. But you have this chunk of the protein that's sitting right smack dab in the middle of the cell membrane, of our bilayer here. And like, well, Mr. Beachka, you just told me that polar and non-polar environments can't exist together. Well, so the way that these proteins can do that is, if you guys kind of recall back to chapter 2 again, remember the amino acids that make up our super long chains of proteins, some of those can be polar and some of those can be non-polar, right? And that creates these areas of the protein that can have polar and non-polar areas. So by definition, this protein, that's the chunk that's stuck right inside here in the non-polar environment between the fatty acid tails, that's a non-polar region of the protein. But then the regions that are outside that are exposed to the external and the internal environment, those are polar regions of those proteins, which is kind of cool, right? So this is one way that we can get around these, like, areas that are completely separate and completely opposite from one another, but we can utilize them to our advantage. So for example, this red protein here is a membrane channel. It is just a tunnel that molecules can pass through willy-nilly whenever they want to. And the way that that is controlled is through concentration gradients. Let's see. This other purple one right over here, this receptor molecule, right? This one has a little chunk down here that becomes activated once a specific molecule is responded to this carbohydrate chain down here, or comes into contact with it. Then this little component down here will then send an entire signal response into the cell and go wherever it needs to go. You can have surface proteins that can also act as kind of receptor molecules to determine whether or not the cell can be identified as a particular cell. So if we talk about COVID for a second, these little surface proteins right here, these guys in green, are an instance of how the cells in our body, our immune system, can detect whether or not a cell in our body has been affected by the COVID virus, right? So what COVID will do is it'll go in and it'll infect the nucleus and make those cells start producing these little surface proteins, right? And that's one of the identifiers of a cell that's been infected by COVID versus a cell that's not been affected by COVID. And like I think we kind of talked about it a little bit, but one of the reasons why you have so much damage to healthy tissue is because the cells that recognize these little bad surface proteins and then neutralize that cell, they're not super specific all the time. So sometimes they end up damaging healthy tissue in the process. That's kind of the downside to that. But putting that in context, these little green guys that exist on the surface of proteins are important because depending on the situation, it's not always the case, but sometimes they can act as identifying proteins for other cells that are floating around to, kind of, it's like their ID badge. Do you belong here? Yes or no, right? So that's kind of what those surface proteins would be used for. Okay. Some other things that we're seeing in here are cholesterol. So if you guys recall, cholesterol is composed of these rings of hydrocarbons that can be shoved into the cell membrane as well. And since it's made out of hydrocarbons, that means that it's non-polar, so it doesn't like to be around water. So it likes to be in where all these other hydrocarbons are, right inside here. So remember, cholesterol is nice because this packed ocean of beads that we're seeing right here, we still want to make sure stuff can get through it if it needs to. And we'll explain a little bit more about that in a second, which is actually kind of the next slide here. But we're going to save that for Thursday. But cholesterol can help with making sure that the cell membrane maintains a certain degree of fluidity, so it can allow certain things through and other things to not pass through. Okay, so what we're going to do is we're going to end a little bit early today because I want to give you guys some opportunities to be able to ask some questions if you have any. I know some of you so Daniel wanted me to talk about some chemical bonds, so we'll talk about those first. But if you guys think of anything, let me know. All right. So let's see here. I'll keep the recording going. So if you guys end up watching this later, you can stop here for the end of our Chapter 3, Part 1 lecture. Okay. So bonds. So Daniel, was there anything specifically you wanted me to talk about bonds, or was there? Oh, what's the YouTube channel? Oh, let me plug my YouTube channel, guys. Was there anything specifically? Oh, so just to review. Okay. Actually, Jacob, while I'm reviewing this, if you go on Canvas, you should be able to find it there in an announcement. So I believe I linked the channel through there. If you can't find it, let me know and I can go back and link it to you. Okay. So chemical bonds. So remember, a chemical bond is a very simple thing. All it is, oh, thanks, Patrick. So all a chemical bond is just a connection between two elements, right? That connection can sometimes be on the same molecule, sometimes it can be between separate molecules. But all it is a connection between two elements, by definition. And for us, there's four that we care about. So Matt, if you go ahead and check right now, there should be some up there. Okay. So for the four that we care about, right? We care about either being able to share an electron or donate them. So chemical bonds can be brought about by either sharing electrons or donating electrons. Okay. So the two sharing ones that are most important are the covalent bonds. And we've got two different kinds. We have nonpolar and polar. The nonpolar ones are where we have even sharing of those electrons between the two atoms. So like a good example of this would be something like carbon bound to hydrogen. Both of those are relatively electrically neutral elements. So the electrons that are shared across the space are done so evenly. So I'll actually use a different color here. Hopefully this one shows up. So those electrons are shared evenly across that space. Let's make this a little bit more apparent here. Let's make this bond a little bit bigger. Those electrons are right in the middle, right? But polar is where they're not evenly shared. So something like oxygen bonded to hydrogen is going to create an uneven sharing of electrons because oxygen is electronegative, meaning that it has a lot of attractive power over electrons. So instead of sharing those evenly, oxygen gets a little greedy, pulls those electrons closer towards its end of that covalent bond. So when we have polar covalent bonds, we create polar molecules. Oxygen's not the only electronegative atom. Things like nitrogen, chlorine, fluorine, phosphorus, those kinds of elements are all electronegative atoms. And if you bind them to something else, you're going to create a polar covalent bond if you can, but then sometimes what will happen is, depending on the situation, you can have something like an ionic bond, where we donate electrons. So a good example of that is our sodium and chlorine reaction. Okay. So since sodium donates its extra, or well, it doesn't have an extra, but it donates its valence electron to chlorine here, they both now have full outer valence shells, but the net positive charge associated with sodium is because it now has more protons in its nucleus than it does electrons, and then chlorine has the negative charge for the opposite reason. Right? So those are ionic bonds. And then the last kind of bond is a hydrogen bond. Hydrogen bonds are interesting because there's no actual sharing or donation of electrons, so they're kind of like in their own category. But hydrogen bonds occur between molecules, so you would never have a hydrogen bond occurring on the same molecule. You'll always find them bonding two separate molecules together. And they occur in polar regions of molecules. Let me make sure that you guys can see that. Yeah, there you go. Okay, so that's the information you should definitely know about chemical bonds. Yeah, absolutely, David. I'm happy to help, man. I'm glad it makes sense. That's good. Good, good, good. Cool. Okay, so let's see if there's anything else. I know I threw around the term acid-base reaction, you guys. Don't worry about that. You don't need to know what an acid-base reaction is. It's not talked about in your book. I'm not going to test you on it. I just threw it out there as like if you were interested in that kind of a chemical reaction, that's what's going on with things like ionic bonds. Let's see. What else? What else? What else? With macromolecules, you should know their monomers or their building blocks. So remember we talked about, like, so there's that big table in chapter 2. Actually, I'll pull that up really quick. Oh, cool, Raina, I'm glad it made sense. So here, let me pull up those slides from last week. So this table here, so table 2.4. If you know the information on this table, you're in great shape with like the second half of chapter 2. Okay. People tend to forget about the building blocks of each of these things. So don't forget about those. Those are important because that's how we build different variations of proteins. It's how we build different variations of lipids or carbs or nucleic acids. Right? Oh, good. I'm glad Quizlet is working for you. That's awesome. Yeah. Let's see. What else? What else? Yeah, pretty much, you guys, with chapter 2 material, the macromolecule stuff, the second half of the chapter, everything, like if you want like a study guide, table 2.4 is it. It's the bee's knees. That's where it's at. Okay, so make sure that you know all of this information in here. So let's move on to like some study strategies. How do you study? How do you prepare for a test in this class? There's a couple different ways you can go about it, but here's what worked for me when I was in your guys' shoes, which wasn't too long ago. But when I was studying anatomy for the first time, actually, I'm going to stop our recording here.